Gazit for the next talk. So the next two speakers are local. I don't need to say much about them. Udi, you know, was our vice president for research and then did some work for the government, and now we have him back to doing real research as a real professor. You're on? Okay.
I'm on. Well, as you know, I did research all the time, but now I have full-time research. So it's really a pleasure and honor to be here. Thank you, Ori and Adi, for the invitation. I'm very happy to be at this part of the symposium because I would like to convince you that we can use organic material, bio-inspired materials, to do things that are similar to what we see with the inorganic materials. And the second part of my talk will be on our work on PNA, which I think it's another branch of, we demonstrated the first time the use of PNA for nanotechnology, and we get some very remarkable structures. Okay, right mode. So our approach for many years already was to use non-biased reductionist approach to look for fundamental molecular recognition and self-assembly modules in nature. We went into smaller and smaller building blocks, as small as dipeptides, which were a working horse for many years. But nowadays we went even to smaller structures, as small as amino acids and nucleobases, which give us new paradigms also for the disease. I will mention it in passing, but more in the context of this symposium, we can make all kinds of architectures at the nanoscale, nanotubes, nanospheres, nanoplates, hydrogels with nanoscale order, and we get unique physical properties: mechanical, optical, piezoelectric, semiconductive, and all with very simple building blocks in which we can control the formation of the structures and their properties. A very interesting point, as I mentioned, our approach is really very systematic. I hope that in the short time that I have, I can convince you, but over and over again, we get into the same solution that nature had provided us with. One very intriguing example for us is what we found time after time, the use of guanine nucleobases, part of the DNA recognition, but also guanine by itself is used in nature for things as diverse as physical colors in fish, the mechanism that allowed chameleon to change their colors, and reflectors in reptile eyes, including crocodiles. We are inspired not only from nature and biological building blocks, but also from polymer chemistry. We are very much into the field of supramolecular polymers. We demonstrate the ability of such simple building block to form ordered supramolecular polymers, and we also bring polymer chemistry concepts into nanoscience and nanotechnology. For example, this quite recent work in which we demonstrated these very simple building blocks. It's Boc diphenylalanine can form all kinds of various architectures at the nanoscale, spheres, fibers, tubes, and we see a typical phase transition and phase separation, as you see with much larger polymers. And we truly could use polymer chemistry concept to understand these activities. I will just mention in passing how the recognition of very fundamental building blocks could lead us to new understanding of biological phenomena. As I told you, by our very systematic approach, we demonstrate that ordered structures that are related to disease could be formed by even single amino acids. This gave a new model for understanding the role of amino acid structures in metabolic diseases and extend the way that we think about the organization of ordered assemblies in metabolic disorders. Yossi Klafter also mentioned our Center for Drug Discovery and related to nanotechnology. I don't have the time to go into details, but by understanding, just to give you the flavor of what you do, by understanding the minimal recognition modules that facilitate the formation of pathological structures, we can design without thinking about the biology, just thinking about the chemistry and way to control the structures. We can manipulate formation of various structures. We already have drug candidates that, based on our ability to understand the formation that completed phase 1 clinical trials. We have other few compounds in preclinical development. And this was recently announced on our agreement to treat Parkinson's disease, the formation of amyloid in the case of Parkinson's disease, just by understanding the concept of molecular recognition and self-assembly. But today I would like mainly to focus on our ability to use, first of all, peptide nanotechnology, and later on PNA, peptide nucleic acid nanotechnology, to get very interesting order. This started in our reductionist approach, trying to identify the smallest recognition module that could allow the formation of amyloid structures related to degenerative disorders. To make a long story short, double meaning, we demonstrated that diphenylalanine, this very simple dipeptide, have all the molecular information needed to form well-ordered nanotubes. We later on, by playing around with the properties, we could get also the formation of nanospheres that are based on the use of this concept, a dipeptide with two aromatic building blocks. We were also able to play around by synthetic chemistry and include a larger aromatic like naphthylalanine, all kinds of substitutions on the aromatic rings like pentafluorophenylalanine to make Teflon-like nanostructures. So we now have a catalog of about 30 or even more building blocks, dipeptide building blocks that can form all kinds of architectures at the nanoscale, which, as I will just mention, have very unique physical properties. So one thing is to make all kinds of different nanostructures. Another thing, and it was referred also in the talk of Chad Mirkin, is to control the formation. And here the simplicity and the minimalism of the building blocks were very helpful for us. So we developed methods including unidirectional growth to make these things like these nanoforests. We use inkjet printing to print these structures, and something that I'm very proud of, and we also got a patent on the concept itself. This was in collaboration with Gil Rosenman. It was the idea of using these building blocks, these biological, bio-inspired building blocks, by utilization of physical vapor deposition. So we can move peptides from the solid phase to the gas phase and completely control the formation of these structures. So this is the electron micrograph with the same magnification of the formation of these structures by self-assembly. We can have either 40 micron nanoforest or 4 micron length, tube length nanoforest, just by controlling the self-assembly of the structures, moving from the solid phase to the gas phase, and then in a cooler substrate on control the assembly. So we can form the structures, we can control the formation. Now for the physical properties. We knew quite from the beginning that these are very rigid structures. But in order to determine really the mechanical rigidity, we had to use AFM. So we're using AFM indentation. So you apply force with the AFM tip on the structures and calculate measure the point stiffness and calculate the Young's modulus. So we could calculate Young modulus for these self-assembled structures built by non-covalent bond from dipeptide. We calculated Young modulus of about 20 gigapascal. For those of you who are less familiar with mechanical rigidity, this is something in the order of bone or teeth rigidity. It's much more rigid than any plastic that we know. This was, at that time, already 10 years ago, we claimed that this is the most rigid self-assembled structures that could be achieved, and nobody reputed it. Actually, independently, a group in Nottingham, Saltender group in Nottingham, calculated the Young's modulus using another method of bending beam, and calculated Young modulus of about 29 gigapascal. Which, if this was a surprise, another surprise came when we wanted to study the nanospheres. So we used the same type of experiment, applying force with the AFM, and nothing happened. Only when we changed the tip, the cantilever, into diamond-based cantilever, we could deform the structures. We measured point stiffness of about nearly 1,000 Newton per meter, and Young's modulus of about 200 to 300 gigapascal. This is like good quality steel, made by self-assembly of dipeptides, no covalent bonds. We are now collaborating with physicists, try to understand the basis for the rigidity of the structures. The advantage of our minimalistic approach is the fact that you can now use not only classical mechanics, but also DFT and quantum mechanics methods to understand it. We still have many mysteries in this, in trying to understand the physical basis for the rigidity. This is why I'm trying to convince more and more physicists to get into this work. Later on, we discovered all kinds, this again in collaboration with Gil Rosenman, all kinds of unique physical properties, other physical properties, piezoelectric properties, similar to the best inorganic piezoelectric material, lithium neobite. Semiconductive properties. I don't have the time to go into the details, but you can find all of this, and I will give at the end a link to my website. Luminescent properties in this of these materials. It's not that we plan to have something that will have these unique properties. Probably not by serendipity, but by very systematic way, we identified a most fundamental recognition model or module that could allow the formation of these structures. And now there are many different applications. Now due to the simplicity, this system is studied by hundreds of groups around the world, and every week I see other applications that were done. Chad Mirkin mentioned a metal organic framework. It was used to get the diphenylalanine in MOFs and use it as energy for the providing translocation of nanoboats. Last week there was some work on the coating of stents with these materials, where use it for synthetic photosynthesis, all kinds of different ideas. Until recently, when I would give such a lecture, people would say, well, it doesn't make sense. What is so unique about diphenylalanine? Yeah, and how can it be that important? And then I would say that it's really in the for our organism like us, it's not very unique. But for things like bacteria, they need some way to control the formation of the structures, and indeed, a few years ago there was funding of an enzyme that all its activity is to cut the diphenylalanine motif, proving the importance of such structures. I will skip this, and diphenylalanine is now studied by many groups. You know that some field is out of your hand when you see such reviews as self-assembly and the application of diphenylalanine nanostructures, and for the first time when it appears in public. So this field became too competitive. We had to switch to another one, and this was the use of PNA for nanotechnology. Our approach, we knew that dipeptides could form ordered structures, so we synthesized all the 16 different combinations of di-PNA. So PNA, it has a peptoid backbone and side chains of DNA and RNA. And we discovered this relates to one of my first slides, that only guanine containing building blocks could form such structures. And I don't have to convince you about the importance of DNA after technology after the talk of Chad Mirkin. When we solved the crystal structure of these assemblies, we discovered that what we had in mind really happened. On the one hand, we had the stacking, as we see with peptide-based structures, but we also get the Watson-Crick interactions between the bases, ended up in very dense materials. And when you get new structures, you get also new physical phenomena. And we could see a phenomena known as red edge excitation shift. This is a phenomena seen in glassy materials, in graphene oxide, in which you have different emission based on the excitation wavelength. Very recently, and just this will be my very last slides, inspired by our ability to show that a single amino acid can form all the structure, we asked ourselves whether a single PNA could form the same assemblies. And again, in a non-biased way, we discovered that only guanine-protected PNA, peptide nucleic acid, can form all the structures. And now see the structure that this very simple building block form. These are uniform assemblies. It looks like silica particles made by the self-assembly. You don't need to do anything. You boil this into the water and cool the solution. We get the spontaneous formation of this array, hexagonal array of these structures, which act as photonic crystals. You see the colors that you see here. During the time that we wanted to publish it, there was a work not from our group, a very nice work from a group in France on the way that chameleons change their color. This is by guanine, our same guanine photonic crystals, and the changing of the spacing is based on the changing of the ionic strength, and thus this you can have a rapid change of color by using photonic crystals. So we can do the same thing with our system. I didn't have a lot of time, and I wanted to show you the different activities. I would like to convince you that that simplicity is the ultimate sophistication. Of course, this is work of many talented students working on these directions and others. And since I moved very fast and showed all kinds of applications that we do in the lab, you're more than welcome to visit my website and see the original papers with the hyperlink.
Thank you very much. So we do have time for a quick question. Yes, Chad, please.
What's the role of the guanine?
The role of the guanine. So,
And why do these structures form such uniform?
okay, this is quite amazing. So the role of it's done only with guanine, and as you know, guanine is used in nature not only in those examples that I mentioned, but also in the molecular level, like G4, quadruplex, and others. This is due to the pattern of hydrogen bonding and the planarity of the molecules that allow the formation of the structure. Why these are uniform structures? I discussed it with experts in the field. Nobody have shown such example with self-assembly of building blocks. It means that probably there's, from the energetic point of view, there's a curvature that is most thermodynamically stable, probably due to geometry, but this is not enough because it's a multi-layer phenomena. So it is probably a balance between the ideal curvature and the entropic penalty that you pay for the formation of the various structures. Actually, we just discussed it with Mike Cates, the Lucasian chair in Cambridge, who told us that he never saw such a system, and it's really intriguing to understand the balance between the energetic penalties of the two structures.
Okay. Thank you very much.